Welcome to the Deep Dive. Today we're tackling a really fascinating stack of research, diving into the incredibly complex world of modern blood banking and transfusion medicine. When you picture donated blood, you probably think, oh, ABO, maybe Rh type. Simple enough. But our mission today is really to unpack that assumption. We want to extract the critical, often surprising details about the huge biological and procedural effort involved. You'll find out that every single bag of blood might just be one of the most rigorously tested, highly engineered things in all of healthcare.
That's absolutely right. That's the core of this deep dive, really. The complexity goes way, way beyond just basic blood typing. To keep a blood supply safe and also, consistently viable for actual life-saving use, the whole system demands this mastery of genetics, immunology, and some pretty sophisticated preservation tech. It's a level of quality control that, honestly, not many other areas in medicine can really touch.
Okay, so let's dig in. Where should we start? Maybe at the very, very small scale. The red blood cell itself. Our sources paint this picture of the RBC membrane as this amazing bit of microengineering. It's a semi-permeable lipid bilayer, okay? But the real structural secret seems to be underneath this mesh-like protein cytoskeleton, like internal scaffolding.
Exactly. And that scaffolding, that structural integrity is, it's everything. A healthy red blood cell normally survives about 120 days circulating in your body. But if there are inherited defects in that structure, either the lipid layer or that protein mesh you mentioned, the cell just can't handle the mechanical stress of pumping around. It breaks down much faster. So keeping that structure perfect is sort of the first big challenge in preservation.
It's kind of amazing that the shelf life of donated blood depends on a microscopic mesh we can't even see. Okay, so from the structure itself to the information on the surface, the antigens, we know the basics, right? Most major blood groups, ABO, Rh, KEL, KID, they follow pretty straightforward inheritance patterns, codominant usually.
Right, that classical Mendelian genetics gave us the foundation. But the field really transformed when we started looking deeper at the molecular level. And what's really interesting is that one of the biggest aha moments historically actually came from studying a blood disorder, sickle cell anemia.
Ah, right. That's where they first really connected the dots, wasn't it?
Sure, precisely. Sickle cell gave us the first hard experimental proof that a specific DNA sequence dictates the specific amino acid sequence of a protein. In that case, it was the beta globin chain coded on chromosome 11. That discovery was fundamental. The molecular blueprint determines the biological reality.
So how does knowing that, having that molecular view, make blood banking better now?
Yeah.
Before, it was all about watching cells clump together in a test tube, right? Agglutination.
Now, we can often bypass that indirect observation and go straight to the source code, so to speak. Modern blood banking relies more and more on molecular methods, things like restriction mapping, PCR, gene sequencing. These supplement, or sometimes even replace, the classic serologic tests, especially for really complex cases. We're not just seeing how the antigen behaves on the cell surface, we're reading the DNA instructions that built it. This is incredibly important for matching patients with tricky antibody profiles.
And that molecular level rigor, it actually starts way earlier, doesn't it? Right at the donation point with a donor safety firewall. Before any blood even gets collected, there are these two key steps. First, the health history questionnaire. It's this standardized form designed to protect both the person getting the blood and the donor giving it.
Absolutely. That questionnaire is like the first filter. It screens very carefully for known risk diseases that could be transmitted through blood. We're talking about things like variant CJD, West Nile virus, malaria, babesiosis, even Chagas disease. It tries to eliminate risks before the donation even begins.
And then, after that detailed history, there's the mini physical. The abbreviated physical exam.
Checking vitals, blood pressure, pulse, temperature.
And crucially, checking the donor's hemoglobin, or hematocrit. Making sure the donation won't harm their health.
Yes, exactly. Once the donor is cleared and the blood is collected, the challenge shifts completely to keeping it viable. Preservation. For standard liquid storage of red blood cells, we can keep them for up to 35 days.
35 days. How?
Well, it's not just refrigeration. It needs a special chemical cocktail, an anticoagulant preservative, usually something called CPDA1.
Okay, CPDA1. That sounds technical. What's it actually doing for the cells for those 5 weeks?
It's doing several things at once, really. The C is citrate that grabs calcium to stop clotting.
Okay.
The P is phosphate, which acts as a buffer to keep the pH stable.
Right.
But the really critical parts are the D-dextrose, which is basically sugar, food for the red cells to keep their metabolism going.
Energy source.
and the A-adenine. Adenine helps the cells regenerate ATP, their main energy currency. Without that precise mix, the cells would just run out of energy and degrade much faster.
Okay, that makes sense. But 35 days is the limit for liquid storage. What about longer term? That's where freezing comes in, right?
Exactly. For really long-term storage, especially for rare blood types, we need to freeze the red cells.
And you can't just stick them in a regular freezer, I assume.
Definitely not. Water expands when it freezes, and ice crystals would shred the cell membranes. So you have to use a cryoprotectant, something like glycerol, in high concentrations. Then store it incredibly cold, think negative 120 degrees Celsius or even colder.
Wow.
Now, freezing is great for archiving blood, sometimes for decades. But the big challenge used to be thawing it out for use. The traditional way involved washing out the glycerol in what we call an open system. That broke the sterile seal.
Meaning it could get contaminated.
Yes, and because of that risk, the thawed red cells were only considered safe for 24 hours.
That's a really short window. How has that changed?
Technology's made a huge difference. We now have automated instruments. You can think of them like sterile washing machines for red cells, a closed system processor.
A closed system, meaning it stays sterile.
Precisely. The whole deglycerolization process happens inside the machine, never exposed to outside air. And if those blood units were frozen quickly after collection, say within 6 days, this closed system processing extends their shelf life after thawing from just 24 hours up to 15 days.
15 days. That's a massive improvement, a real game changer for managing rare blood inventories or emergency stocks.
Absolutely.
Now, speaking of storage challenges, platelets are completely different, aren't they?
Right.
We keep red cells cold or frozen, but platelets, they're stored warm, like room temperature, 20 to 24 degrees Celsius with constant shaking, and only for 5 days max.
That's right. And that warm storage, that's the key issue. It makes platelets much more vulnerable to bacterial contamination than refrigerated or frozen red cells.
Because bacteria like warmer temperatures.
Exactly. It's a much friendlier environment for any bacteria that might have gotten in during collection.
So if you can't use cold to stop bacteria, how do blood banks deal with that risk for platelets?
There's a really clever, relatively simple procedural fix: diverting the first bit of blood collected.
Diverting? How does that help?
Well, when the needle first goes into the donor's arm, it can sometimes dislodge tiny bits of skin plugs. And those skin fragments are actually the most common source of bacterial contamination getting into the collection bag.
Ah, I see.
So by collecting the very first 20 or 30 milliliters of blood into a separate small pouch and then discarding it.
You divert the potential contamination away from the main product.
Exactly. Studies show this simple step can reduce bacterial contamination in the final platelet unit by anywhere from 40% to even close to 90%. It's a very effective safety measure.
Okay, so the blood is collected, preserved. Now it needs to be matched. This is where the high-stakes immunology comes in, right? The testing. We often hear about antibody reactions being like a lock and key, but our sources say whether that lock and key actually connect depends on a lot of factors.
Oh, absolutely. It's very sensitive. Things like the distance between the red cells, the concentration of antigen versus antibody, the pH of the solution, the temperature.
Temperature matters.
Big time. And critically, the type of antibody involved, whether it's an IgM or an IgG immunoglobulin.
Okay, why does the antibody type IgG versus IgM matter so much clinically?
Well, IgM antibodies are typically large molecules. They tend to react best at colder temperatures, like room temp, often showing up right away and testing what we call the immediate spin phase. But usually these IgM reactions aren't clinically significant, meaning they don't cause major problems if transfused.
So they're easier to detect but less dangerous.
Generally, yes. The ones we really worry about are the IgG antibodies. These are smaller, they react best at body temperature, 37 degrees C, and they are the ones usually responsible for serious transfusion reactions.
They're harder to find.
They often require an extra step in testing called the anti-human globulin phase, or AHG phase, to become visible.
Wait, why is that extra AHG step needed? If the IgG antibody binds to the red cell antigen, why doesn't it just cause clumping like the IgM does?
That's a great question. It comes down to size. IgG antibodies are just too small. Even when they bind to antigens on two different red blood cells, they can't physically span the distance between those cells to pull them together into a visible clump.
They're like tiny little bridges that are too short.
Exactly. So the AHG region is basically an antibody against human antibodies. It's designed to bind to those IgG antibodies that are already stuck on the red cells. The AHG molecule acts as a larger bridge, linking the IgG-coated cells together, and that finally causes the visible agglutination, the clumping, that tells us there's a clinically significant antibody present.
That is really clever,
Yeah.
like building a bridge extension. Now, to handle really complex antibody situations, the sources mention something that sounds, well, almost counterintuitive, using enzymes like physin or papain. And these enzymes actually destroy certain blood group antigens on the red cells, like Phi, Phyb, M, N, and S.
Yes, they do. It sounds destructive, but it's actually a very powerful diagnostic tool because it's selective. While those enzymes are destroying antigens like Duffy, Phi, Phyb, and MNS, they simultaneously enhance the reactivity of other antigens, specifically Rh antigens, KID, P1, Lewis, and I. Antibodies tend to react stronger after enzyme treatment.
Whoa! So it knocks out some signals and boosts others.
Precisely. By comparing reactions with untreated cells versus enzyme-treated cells, you can start to piece together which antibodies might be present in a patient's plasma. If a reaction disappears after enzyme treatment, it might be Duffy or MNS. If it gets stronger, it might be Rh or KID. It helps isolate the culprits.
It's like using chemical scissors and amplifiers.
Yeah.
Very cool. Okay, let's connect some of this back to genetics and, well, human history. The Duffy blood group system comes up again. The Phi AB phenotype lacking both main Duffy antigens is apparently very common in black populations, but almost never seen in white populations. Why the difference?
It's one of the clearest examples we have of natural selection acting on human blood groups. The Duffy antigen, the protein on the red cell surface, is actually used by the malaria parasite Plasmodium vivax to get inside the red blood cell.
No way. The parasite uses the blood group antigen as a doorway.
Exactly. So individuals who genetically lack the Duffy antigen, the Phi AB phenotype, are naturally resistant to infection by P. vivax malaria.
Wow. So in areas where that type of malaria was widespread, like parts of Africa, having that Phi AB blood type was a massive survival advantage.
A huge advantage, which explains why that specific genetic variation became so common in those populations over generations. It's evolution written in our blood cells.
That's incredible. Okay, shifting back to the lab bench and the challenges blood bankers face. One system that gets mentioned as particularly tricky is the KID system, JK antigens. Apparently, KID antibodies are notorious for causing delayed hemolytic transfusion reactions. Why are they so difficult?
The main problem with KID antibodies is that they have this tendency to, well, fade away. They often disappear pretty rapidly, both in the patient's bloodstream, in vivo, and even in stored blood samples in the lab, in vitro.
They just vanish. So someone could have had a reaction, develop the antibody, but later test negative.
Yes, that's the danger. A patient might have had a KID antibody detected months or years ago after a transfusion or pregnancy. But when they come in for a new transfusion, their current antibody screen might be negative because the antibody level has dropped below detection limits.
So what happens if they get transfused with KID-positive blood based on that negative test?
The transfusion acts like a booster shot. The patient's immune system remembers that KID antigen, and it quickly ramps up production of the KID antibody again.
But not immediately.
No, typically days later. That's why it's a delayed hemolytic reaction. The newly formed antibodies start destroying the transfused red cells, often after the patient has already gone home from the hospital. This highlights why checking patient history for previous antibodies is just as crucial as the current testing.
That sounds incredibly important to track. Okay, one last area where there seems to be some common confusion. Hemolytic disease of the fetus and newborn, or HDFN. Let's contrast the two main forms.
Right. ABO-HDFN and RH-HDFN. Clinically, they are very different beasts. ABO-HDFN happens when a mother, usually type O, has IgG antibodies against A or B antigens that cross the placenta and affect her type A or B baby.
Okay.
But while it's relatively common, severe problems like serious fetal anemia are actually extremely rare with ABO-HDFN. The most common issue is jaundice, hyperbilirubinemia after the baby is born, which is usually manageable.
So, common but generally mild. What about RH-HDFN?
RH-HDFN, classically caused by a D-negative mother developing antibodies against her D-positive fetus, is historically much more severe. Before we have effective prevention like Rh immune globulin, RH-HDFN could cause devastating fetal anemia, hydrops fetalis, really serious complications, often requiring interventions even before birth.
So the RH type is the one that really needs the careful management, like giving rig shots.
Exactly. While both are types of HDFN, the clinical severity and management are worlds apart. It's important not to conflate the two. Hashtag outro.
So after diving through all this, when you really step back and look at everything involved, from checking a donor's iron levels to understanding the protein scaffolding inside a single red cell, to using enzymes to enhance some antibody reactions while destroying others. It really hits you. Managing just one single unit of blood safely requires this almost unbelievable depth of very specific knowledge.
It really does. And the constant theme throughout the history of blood banking has been this relentless push for greater safety. A huge leap forward in that regard was the introduction of nucleic acid testing, NAT, for donor screening.
Right. Instead of looking for the body's response to a virus, the antibodies, NAT looks for the virus's genetic material itself.
Exactly, which means you can detect an infection much, much earlier during the window period before antibodies even develop.
And the impact that had was just massive, wasn't it?
Absolutely staggering. For HIV, the infectious window period with the older antibody tests was around 22 days. NAT slashed that down to about 11 days.
Cut it in half.
And for hepatitis C, HCV, it was even more dramatic. The window went from roughly 82 days down to just 10 days with NAT. Think about the infections that were prevented by closing those gaps.
It's incredible. Molecular biology directly translating into saving lives on a huge scale.
Yeah.
Okay, so thinking about that trajectory, the increasing precision, moving from visible clumping to reading the DNA itself.
Yeah.
Here's a final thought for you, our listener, to chew on. Given this ever-deepening understanding of genetics and immunology and blood groups, how might future technologies change things even more?
Yeah, think about things like rapid, maybe even point-of-care, whole genome sequencing, or perhaps nanotechnology applied to blood compatibility or even synthesis.
Will we eventually reach a point where blood safety isn't just about screening, but about designing or modifying blood components tailored perfectly to the recipient's unique molecular profile? What will the outer limits of blood safety look like in the next decade or two?